everyone, this is Fiza from Group 316. Today's my topic is replication of DNA. One cell division, the DNA material is doubled and there occur the reduplication of the DNA double helix. In principle, three mechanisms of replication are possible. Conservative replication, with the original DNA double helix remaining intact and a new daughter double helix being formed. Semi-conservative replication, in which two daughter double helix contain each one new and one old chain. Finally, the dispersion mechanism involves the distribution of original material among the four chains of the two daughter double-stranded molecules. Melsons and Stahel studies the replication of DNA during cell division of E. coli with the aid of labeled atom and sedimentation in the cesium chloride density gradient. They produce a population of 15N labeled cells. These cells were transferred took place from the original population and from that of first and second generation molecules. There were isolated DNA molecules and their density and radioactive width determined. The parental DNA molecule labeled with 15N had the largest density. The DNA of the first generation was found to be half labeled and its density was equal to the arithmetic mean of the densities of the 15N and 14N labeled DNAs. Finally, the DNA of the granddaughter molecule was separated on the sedimentation into two zones. One zone with DNA being identical with the daughter molecule and the other zone with unlabeled DNA. This may be depicted schematically as follows. The newly formed DNA has the biological properties of parental DNA. For example, synthesis with the DNA from phage alpha into 174 as the primer produce DNA which has a infective stage. Phage particles are synthesized using a polymerizing enzyme, DNA polymerase. One can produce synthetic polydeoxyribonucleotides capable of forming double helix in the presence of complementary. The free energy change during the formation of two double helix from one parental double-stranded DNA and nucleoside triphosphate may be represented in the following form. The unwinding of the DNA double helix on replication must presumably be different from the involved in denaturation. The gain in free energy during the formation of new chain is responsible for a high angular momentum. New DNA is synthesized in vitro always in the 5 to 3 direction. At the same time, there are data which indicates that the two chains are replicated, one in the 5 to 3 direction and another in the 3 to 5 direction. Okazaki ascribed this is the discontinuous mechanism of replicative synthesis. Short sections of the new DNA are synthesized on one or both chains at the replicating point, invariably in the 5 to 3 direction. Then these sections are joined forming new chain. If model is correct, the last replicated section of one or both daughter chains can be separated. The temporary selective inhibition of enzymes that catalyzes the accumulation selective inhibition of the enzyme that catalyzes the accumulation of phosphodiester linkage between the DNA chains. These predictions are verified by experiment which shows that the tritium labeled thymidine is inserted into the short chain which only after a while unit in the long ones. Steps of DNA replication. Initiation. Helicase unwind DNA and primase add RNA primase. Unwinding. Helicase separate DNA strands. Synthesis. DNA polymerase matches nucleotide to template strand. Elongation. DNA polymerase extends new DNA strand. Proofreading. DNA polymerase correct errors. Ligation. DNA ligase seals gaps between nucleotides. Complications. Completion. Replication is completed and DNA is duplicated. Key enzymes: helicase, primase, DNA polymerase, DNA ligase, topoisomerase. Error and repair. Mutation, base substitution, insertion, deletion. Proofreading and editing. DNA repair mechanism: base insertion repair, nucleotide excision repair. Importance of DNA replication. Genetic inheritance, cell division and growth, evolution, challenges and diseases. Genetic disorders, example cancer, inherited disease. Replication stress, DNA damage and repair defects. The next topic is mutations. Mutation occur either spontaneously or under the influence of external factors, chemicals, or radiation effects on chromosome and genes. The following two types of mutation should be distinguished. Chromosomal mutation, reconstitution of the chromosome observable under the microscope, and point mutation. There exist four types of mutation. Point mutation, single nucleotide chain. Frame shift mutation, insertion or deletion of nucleotide. Chromosomal mutation, change in chromosome structure or number. Genomic mutation, change in entire genome. Spontaneous gene mutation are determined by error in DNA replication that arises because of the thermal motion of atom and molecules. Evidently, these errors of transcription and translation are not inherited. The insertion of the nucleotide that is not complementary to the template nucleotide. The deletion or replacement of the nucleotide usually led to the formation of loop in the DNA double helix. In subsequent replication of the DNA loop disappears as a result of the semi-conservative synthesis, but the primary structure of the DNA remains unchanged. The formation of loop may be accompanied by the formation of a pair different from a Watson-Crick pair due to the ability of a nitrogen base to form unusual hydrogen bond, and also due to the tautomerism. The replacement of the pair adenine thymine by guanine cytosine is uh, evidently thermodynamically favorable, since guanine bonds to the cytosine more strong than is adenine to thymine. It were for thermodynamics alone, the relative guanine and cytosine content in the DNA should have increased during the evolution. This is not the case in actuality. In higher organisms. The guanine cytosine content is stabilized at 40 to 45% level. The evolutionary formation of a guanine plus cytosine organism is biologically meaningless because triplets that do not contain adenine and thymine codes only for proline, arginine, alanine, and glycine. That is only 1/5 of all the amino acid residues. The replacement of nucleotides can be divided into two groups. First, transition, and second, transversion. Transition is a point mutation in either DNA or RNA is in which there is a replacement of one purine by another or a replacement of one pyrimidine by another. The transversion or point mutation in either DNA or RNA in which there is replacement of purine by pyrimidine or vice versa. We have considered missense mutation which leads to the replacement of an amino acid residue. We shall now turn to nonsense mutation which leads to the termination of the protein chain. There are altogether 23 such mutations which involve single nucleotide replacement in codon XYZ, 9 replacements in X, 7 replacements in Y, and 7 replacements in Z. These 23 mutations, 18 are transversion and only 5 are transition. The lower probability of transversion lower to a certain degree the probability of lethal nonsense mutation. Cause of mutation: error during DNA replication, environmental factors, example radiation and chemicals, viral infections, genetic recombination. Mutation classification: germline mutation inherited, somatic mutation acquired during lifetime, driver mutation contribute to disease or cancer, passenger mutation no functional changes. Mutation effects: silent mutation no noticeable effect, missense mutation altered protein function, nonsense mutation premature stop codon. Loss of function mutation reduce or eliminated gene function. Gain of function mutation increased or new gene function. Disease associated with mutation. Genetic disorder, example cystic fibrosis, sickle cell anemia, cancer, neurological disorders, example Alzheimer, Parkinson's, infection disease, example antibiotic resistance. Mutation detection methods: DNA sequencing, PCR, polymerase chain reaction, genotyping, next generation sequencing. Mutation repairs mechanism: base excision repair, nucleotide excision repair, mismatch repair, homologous recombination. Current research: personalized medicine, gene editing, CRISPR-Cas9, cancer genomic synthetic biology. Thank you.